Alright, so today we're going to look at how we can measure the cell under a microscope. So before that, let's look at the microscope, just a review. Okay, so this is the microscope that you are familiar with, that you have used in the lab for your practical lessons already. Okay, just to do a little bit of recap, making sure everybody knows the structure and the parts of the microscope. Okay, so you have here, that's the eyepiece. Okay, and then you have your objective lens. And then your stage. So, if you notice, or if you remember, when we did our practical, when you look into the eyepiece, you will see something that looks like this. Okay, so what you're seeing, this one right now, okay, you have this transparent ruler, right? Now that is known as the eyepiece graticule. Okay, I'm just gonna write the words, the name for you. So this is the eyepiece graticule. Right, this is what you can see as you're viewing into this eyepiece, right? Now notice that it has sort of like a mini ruler inside it, and then you can see that it has numbers from 0 to 10, and if you actually count the divisions, I'm just going to zoom that in a little bit. If you actually count the divisions, there are actually a total number of 100 total divisions. Okay, just try that one more time. See, there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So that's like 10. There's a total number of 100 divisions. Okay, what we need to do is we need to be giving the numbers to these divisions. Okay, now, so when we're trying to give the units to this eyepiece, okay, what we're trying to do is we need to calibrate. So the term is to calibrate the eyepiece graticule. So calibrate. All right, we will need a special second set of ruler. All right, now what is that second stage of ruler known as? It's the stage micrometer. Okay, so what you can see here in front of your screen, okay, this is a stage micrometer. So I'm going to zoom it in a little bit. It's called the stage micrometer. Is that on the stage, what you will place is here on the slide. Notice that there is a tiny circle in the middle. All right, what that would look, if we try to zoom it in a little bit, is that it will have something that looks like this. In other words, it's kind of like another set of ruler, right? But the difference between the stage micrometer and the eyepiece graticule is that we know exactly the length of this stage micrometer. All right, so if you look here at the information at the bottom, okay, this is a ruler with 100 divisions. So that's what it means. It has 100 divisions, right? Each of that division is actually 0.01 millimeters, right? So the whole length is 1 millimeter. All right, so just to re-emphasize, to clarify, all right, I'm just going to draw another ruler. I'm trying to make it as thin as possible. Okay, notice that I've drawn here the line in purple. That means for this 100 divisions of the stage micrometer, this whole length from this end to the next end is actually 1 millimeters, which they have stated, right? I'm actually going to highlight that in the same color so that you remember that I'm referencing to that number. And they're very nice to you that they have done, or they've told you that that means each of this division right, is 0.01 millimeters. So it's each of this division. I'm going to try to zoom it in a little bit further for you guys. Now, unfortunately, I can't really zoom in any much further than that. But yes, okay, look here. If you try to bring your attention towards this one division, so that length in blue is 0.01 millimeters. Now, let's do a quick recap of how you are going to convert the units from millimeters to micrometers. All right, just do a quick recap, unit conversion. From millimeters to micrometers, okay, what you need to do is you need to multiply the value to 1000 or times 10 to the power of 3. Okay, so with that information that you have, right, since we know that each division is 0.01 millimeters, okay, one division, so one stage micrometer unit or stage micrometer division is equals to 0.01 millimeters. That means that would be equals to 0.01 times by 1000. You can check with your calculator. So 0.01 times by 1000, that would be equivalent to 10 micrometers unit. Okay. Oops, okay, just gonna. Okay, so how is that going to be useful? So this 10 micrometers unit, we're gonna use this value that we know, the division unit, to calibrate the eyepiece graticule. How do we do that? So the first thing we are going to do is we are going to put this stage micrometer and this stage micrometer onto the stage of our microscope. Okay, I'm gonna put them here. I'm gonna put this here on the stage of the microscope, and we are going to align them with our eyepiece graticule. Okay, so we're gonna align them, and I'm gonna use an image for an example. Okay. So what you're seeing now is an example of how we can align, okay, the stage micrometer and the eyepiece graticule. So micrometer or graticule, yeah. All right. Now, don't panic. I know that you noticed that, oh, why is it there's 100 here, but in the earlier picture, I use 0 to 10. It's okay, right? So what you can do is, you can just use the numbers and read it accordingly. Okay, for this particular eyepiece graticule, okay, I'm just gonna put this one, micrometer, also the eyepiece graticule. All right, so this one has 0 to 100. Makes it easier. What it means is that this one is just 10 divisions, 20 divisions, 30 divisions, or 10 eyepiece unit. All right, oops, sorry, this one would be 10 eyepiece unit, 20 eyepiece unit, okay, unit or division, okay, which one you're comfortable with can be used. All right, so what must you do? The first thing that you must do is you are going to try to align the stage micrometer at the bottom and your eyepiece graticule together. Noticing that in this image, what they have done is that they had tried to align the stage micrometer to be exactly here at 0, aligned together at 0 units of the, or 0 division of the eyepiece graticule. Right? And the next thing that you will need to do is you need to find the next line where the stage micrometer and the eyepiece graticule are well aligned to one another. Okay? Right? You're going to have to be looking, since we know that on the left side, because it's like the thicker part of the stage micrometer, towards the left. So try to find the next division where it aligns, the top and the bottom are exactly aligned with one another. So this one could be another example. Okay, that's one. But you don't necessarily have to choose that one. You can pick the next closely aligned, well-aligned divisions together. This one is not quite. Notice he's going to look at the left side of the line, right? So that's not aligned well. Okay. Let's see this one. This one could be. Can use this one as well. Yep. Right. The further it is, the better. Okay. Actually, why can we do this? Do they appear to be aligned? Let's see. Not quite. Okay, if you bring your attention here, I noticed that that is what I'm going to pick. It seems to be the best aligned, right? Ujung-ujung at the ends. Towards the left, these two are best aligned. All right, so we are going to go through the measurements together. Okay, so reading through this image that has been provided. So let's look at the top. So let's measure how many eyepiece unit is that. So that is 65, 66, 67, 68, 69. So that is 69 eyepiece unit. 69 eyepiece unit is equivalent to 10, 20, 30, 40, 46. So that is 46 stage micrometer unit. So that is 46 stage micrometer unit. Remember that each division is mentioned here. It's 0.01 millimeters, which is, we've already calculated in the earlier examples, right? This is equals to 10 micrometers. So 46 stage micrometer units times it by 10 micrometers is equals to, you can do the calculation and check, that should be equals to 460 micrometers. So since we want to find out how much exactly is this one division of the eyepiece graticule, what we can do is, if it's 1, how much is that? So what you can do is, you can just divide the value. This is a cross multiplication. Therefore, 1 is equals to 460. Divide that with 69. Okay. Then you can do your own calculation and check. What by 69? That would be 6.67 micrometers. Okay. Now, what can we use that information? Or how can we use this information that we have just obtained here? Hold on. Now that we have the information of what one eyepiece graticule division or unit is equivalent to, if you look at the bottom, what I have put is, let's just say we are viewing a cell under the microscope, and this red cell, right, I've placed it under the microscope, and we can view it under with the eyepiece graticule. What you can do is you can actually measure that length of the cell. So in this case, well, just to make our example easy, notice that the length or the eyepiece unit of the cell is between 40 to 60. That means 60 minus 40, that's equal to 20 eyepiece unit, right? That is 20 eyepiece unit. We know that one eyepiece unit is equals to 6.67. Therefore, for this particular example that I have chosen here and drawn purposely for the sake of our exercise today, is that what we can do is that we can just times that with our calibration values. Okay, 6.67. So that means 20 eyepiece unit times by 6.67 micrometers, that would give you the value of the length of this particular object. So that is equals to 133.4 micrometers. Which means then the length of this object, okay, is 133.4 micrometers. Okay, so that's how we give a value to the divisions of the eyepiece graticule that we see under the microscope. Okay, now that you have done the first example, what you need to know is that you will have to repeat the calibration process for each of the magnifications of the objective lens. You would first do it for ×4, record the value for one eyepiece unit after calibration, do the same one at ×10, do another one at ×40, and you will keep this value. All right, the calibration value will be kept. Okay, so you can use the calibration value. So you can use the calibration value every time you measure with the microscope. But remember, if you use a different microscope, okay, you have to calibrate them again. Because each microscope have different magnification. Okay? So this is an example of how we might be asked about calibration. This is from paper 1. So the diagram shows a stage micrometer with divisions of 0.1 millimeters apart, viewed through an eyepiece graticule. So hopefully you can recognize that at the top, that is the stage micrometer, at the bottom is the eyepiece graticule. Now they told us already of the value of each division. So that means each division here is equivalent to 100 micrometers. So let's calibrate that first. Okay, so we have eyepiece unit with stage micrometer values, right? So we have 40 eyepiece units, which equals to 100 micrometers, which is 1 SM unit, right? So 1 eyepiece unit would be equals to X. So what you can do is just cross multiply 100 divided by 40. That should give you 2.5 micrometers. Okay. But we are asked to find the area of the field of view. Now, what is the field of view? Is the view that you can see here under the microscope. All right. And then because this is a circle, the formula of a circle is pi r squared. And you need to find the radius. So in this case, the radius would be half of a diameter, isn't it? And we can see from the eyepiece graticule, half of this field of view is 50 eyepiece unit. Therefore, to find the radius is equals to 50 eyepiece unit times by the calibration value that we found here times 2.5 micrometers, okay, equals to 125 micrometers. Now, the third step is we can just apply the formula pi r squared, so 3.14 times 125 micrometers squared. Okay, so that's 49062 4.5 micrometers. Okay, now if you look at the options at the bottom, okay, we have various answers in standard form. So the correct answer here would be D. Okay, what we can do is, if you can try and do that, I think everybody should be able to do this by now. So if you move over the circle, it's 1, 2, 3, 4. So 4 jumps, so that is equivalent to 4.91 times 10 to the power 4 micrometers squared. Oops, I forgot the square units here. There you go.